All right, everybody. This is the Ghost of Kennewick Man Part II. The Ghost of Kennewick Man Part II. You know, hopefully by this time you guys would have tuned in to Part One and gotten the information leading up to Part Two. Which in Part One I explain the background, the professional background of one of the geneticists that is a part of the study that I'm going to read. And in this segment of part two, I'm gonna get into the second geneticist that's a part of this study. They partnered together on this study. And I'm gonna get this music. Yeah, to set that vibe up. Let me take it down a little bit. Make sure I'm trying to be up. That's good enough. So yeah, that's what I'm gonna do. And let's clap it up for this groundbreaking study, man. Let me get some hand clapping. Let me get another one. But anyway, just having some fun here, man. But to get down to it and to get serious, I'm gonna start off with the background information of the second geneticist, and I'm gonna pull that up by sharing my screen. And so yeah, that's what I'm gonna do. Yeah, this is it right here. So I've already had it pulled up already. And so let's just go through it real quick. I asked, who is Shi Huang in the scientific realm? It answers by saying, in the scientific realm, Shi Huang is a molecular geneticist, evolutionary biologist, and professor at the State Key Laboratory of Medical Genetics at Central South University in Changsha, China. He is best known as the primary architect of the maximum genetic diversity hypothesis, which I explained in detail in part one. A controversial and unorthodox framework that, that challenges some of the most fundamental decades-old assumptions in evolutionary biology. The core of his research, the MGD, hypothesis. Most of Shi Huang's academic footprints centers on dismantling the neutral theory of molecular evolution, which once again I explained in part one. The mainstream view. Mainstream science believes that the vast majority of genetic mutations are neutral, neither helpful nor harmful, and accumulate linearly over time. This creates a molecular clock that scientists use to calculate when two species split from a common ancestor. Shi Huang's view, Hong argues that this framework is a mathematical illusion. Huang argues that this framework is a mathematical illusion. He's calling the current framework a mathematical illusion. His MGD hypothesis posits that the genetic diversity does not grow indefinitely over time. Instead, he argues that the population maximum genetic diversity is strictly capped by a physiological ceiling determined by environmental selection and organismal complexity. Once a species hits this ceiling, its DNA enters a saturated mutation phase, completely scrambling the molecular clock. So based on what I read right there, it seems like it's a possibility that, coalescent times, may be misattributed, because he's suggesting that once it hits this saturation phase, it's very difficult to tell apart or identify coalescent times because they're so scrambled. But moving on, unorthodox views in human origins and migrations. Using his MGD framework to filter out saturated genetic noise, Wang, collaborating with fellow researcher Beijing Wan, has published papers and preprints that attempt to rewrite human prehistory. His findings radically clash with mainstream scientific consensus. In the East Asian, let me make sure I shared this on the screen. Now it's on the screen. I'm glad I checked that, man. So let me go back up again and read it to make sure you guys can see it. Who is Xing Hu Wang in the scientific realm? As you can see here, it reads, in the scientific realm, biologist of medical genetics at Central South University in Changshan, China. He is best known, and let me do this as well. He is best known, wrong tab. Had to make sure I ain't make no more mistakes. Make sure my mic is on. But anyway, he is best known as the primary architect of the maximum genetic diversity, MGD, hypothesis, a controversial and unorthodox framework that challenges some of the most fundamental decades-old assumptions in evolutionary biology. The core of his research, the MGD hypothesis, most of Xing Hu Wang's academic footprint centers on dismantling the neutral theory of molecular evolution, which was originally proposed by Pt II in the late 1960s. Let's see let's see if I can copy and paste that name. And I'm gonna bring up a different window. We can see who he is. I just want to get a little bit of background on him. I ain't gonna make it complicated to spend too much time on it. Ooh. Tell me a little about So AI is saying that he was a brilliant Japanese biologist who fundamentally revolutionized our understanding of how evolution works at the microscopic level. He is widely regarded as one of the most important evolutionary geneticists of the 20th century, with some peers ranking his impact alongside that of Charles Darwin. Here is an overview of his life and groundbreaking work. The father of neutral theory. Before Kimura, the scientists' consensus was firmly pan-selectionist, meaning biologists believed among almost all evolutionary changes were driven by neutral selection. In 1968, Kimura turned this consensus on its head by introducing the neutral theory of molecular evolution. His theory proposed that genetic drift over selection at the molecular level, DNA, RNA, and proteins, the vast majority of the evolutionary changes and genetic variations are not caused by Darwinian natural selection, Instead, they are caused by the random fluctuations of mutant genes that are neutral, meaning they neither help nor harm the organism's survival. Evolution by chance. Because these mutations have no biological advantage or disadvantage, they spread through a population purely by random chance sampling from generation to generation. The degenerate code. This happens largely because the genetic code is redundant. Many distinct DNA mutations result in the exact same amino acid or proteins, remaining completely invisible to the forces of natural selection. Remaining completely invisible to the forces of natural selection. Note, Kimura did not reject Darwin's theory. He agreed that natural selection shapes an organism's visible physical traits. However, he proved that the baseline molecular level, random chance rules supreme. That at the baseline molecular level, random chance rules supreme. Okay, just wanted to get a little background on that, man. Anyway, we'll get back to this here. The mainstream view. Science believes that the vast majority of genetic mutations are neutral and accumulate linearly over time. This creates a molecular clock that scientists use to calculate when two species split from a common ancestor. So once again, obviously, Shi Huang's model is contradictive to this linear model, which they're probably, in my opinion, they're probably using that model to basically force feed narratives, man. And this article is going to go into detail in terms of why and how. So, Shi Huang's view. Huang argues that this framework is a mathematical illusion. His MGD hypothesis posits that genetic diversity does not grow indefinitely over time. Instead, he argues that a population maximum genetic diversity is strictly capped by a physiological ceiling, determined by environmental selection and organismal complexity. Once a species hits this ceiling, its DNA enters a saturated mutation phase, completely scrambling the molecular clock. Unorthodox views on human origins of migrations. Using his MGD framework to filter out saturated genetic noise, Huang, collaborating with fellow researcher Dewan Wan, has published papers in preprints that attempt to rewrite human prehistory. His findings radically clash with mainstream scientific consensus. An East Asian origin for modern humans. While mainstream genetics overwhelmingly support the recent out of Africa model, Huang has used his alternative genetic models to argue that modern human Y chromosomes and mitochondrial DNA actually originated in East Asia, dispersing elsewhere through hybridization with archaic humans. Now, in previous episodes, and I've actually argued with other people, like the dude Sean P. You can find that, back and forth with him. When, I argued in my episode, African Americans are not West Africans, that there has been suggestions by multiple geneticists to suggest that the lineage associated with so-called African Americans, which is the E lineage, which is associated with, on average, I think it's like between 70 and 80% of most African American males. That lineage was a part of a superclade called the DE superclade. Now, what made it so controversial is because the sister lineage, which is D, is mainly found in East Asia, which puts a stress on the out of Africa model, because you have two different lineages on two different sides of the landmass. Really, Africa is not considered to be part of the Eurasian landmass, but you guys get what I'm saying. Okay, you have, based on the out of Africa model, you have E in Africa, and you have the majority of the D lineages in East Asia. And with those particular East Asian D lineages not having any fossil evidence between Africa and East Asia, which makes it almost impossible for that D lineage to originate in Africa and then migrate to East Asia. So in my conversation with Sean P, we both found that Michael Hammer determined that it was a toss-up, that he couldn't determine whether that DE split in Africa or whether the DE split in East Asia. And here it is, we have another geneticist, two of them, indicating that they believe that the origin of mankind began in East Asia. Because, as I've said before, I can't remember if I actually said it on an episode of one of my YouTube episodes, is that the out of Africa theory did not solidify until 1930. When it was first proposed, it had heavy scrutiny and it was not widely accepted until about 1930. Previously to that, the origin of so-called Negroes was predominantly thought to be from East Asia. I'm not saying I believe that that's their understanding, but what I am saying is that the out of Africa model wasn't the original model proposed for the origin of any Negroes. So I'm just putting that out there. Reviving Alternative American Migrations. In his co-authored paper on the peopling of the Americas, molecular evidence for the Paleo-American and the Sotrian models, Wong used his slow S&P method to analyze ancient genomes like Kennewick Man and Anzac 1. He concluded that the data validates alternative migration theories, such as European Ice Age hunters crossing the Atlantic, the Solitarian hypothesis, and early Australo-Melanesians populating South America, which is consistent with the study about the Australian signal. And I might highlight that again just to make sure I hammer home what that paper is actually saying, because the Australo-Melanesian signal in the Amazon is actually saying that that signal is not found in other Native American groups. Upstream, throughout Central America, North America, it's not found in Alaska, it's not found in Europe, it's not found in Asia either. And it's not even found amongst the modern Australo-Melanesian populations today, which is very interesting. Okay, I'm just going to read this summary and then I'm going to get on with the actual paper itself. In summary, the scientific community, in the scientific community, Xu Wang occupies a unique dual space. To mainstream evolutionary biologists and anthropologists, he is viewed as a fringe contrarian whose alternative genetic theories rely on non-standard interpretations of genomic data to support discarded historical models. However, in the field of medical genetics, he is recognized as a legitimate quantitative researcher working to decode the genetic underpinnings of complex hereditary diseases. So basically, it is saying like this, man, when it comes to, overriding our standing model, you're fringe. We'll accept your other work in the medical field, man, because you know what you're doing. You're an expert on that end, so we gotta accept that because you're benefiting the medical field. But we're not going to give you any credit pertaining to your model as it pertains to how to analyze S&Ps. That's cool though, because nonetheless, at least in my opinion, it is what it is. And this article that we're about to get into is gonna be a head cracker, boy. So let's do it. Let's do it. Here we go. Let me go back to the top. Yeah, here we go. So you guys already know who the author is. It seems like this was published on May 1st, 2017. You can see the institutions that published it. The article is titled "On the Peopling of Americas: Molecular Evidence for the Paleo-American and Soltrian Models." A Soltrian model was explained in part 1. You can go check that out if you're not familiar with it. Probably be a lot of people who are already familiar with it, independent of part 1. But anyway, we'll read this abstract. Morphological and archaeological studies suggest that the Americas were first occupied by non-Mongoloids with Australo-Melanesian traits, which was subsequently followed by Southwest Europeans coming in along the pack ice of the North Atlantic, and by East Asians and Siberians arriving by the way of the Bering Strait. Yeah. Past DNA studies, however, have produced different accounts. With a better understanding of genetic diversity, we have now reinterpreted public DNA data. Consistent with our recent findings of close relationship between South Pacific populations and Denisovans or Neanderthals who were archaic Africans and Eurasian admixtures. The 9,500-year-old Kennewick Man skeleton with Australo-Melanesian affinity from North America was about equally related to Europeans and Africans, least related to East Asians among present-day people, and most related to the 6,500-year-old Neanderthal Mezhmaiskaya 1 from Adygea, Russia, among ancient Eurasian DNAs. The 12,700-year-old Anzac 1 of the Clovis culture was most related to the 18,720-year-old El Moran of the Magdalenian culture in Spain among ancient DNAs. Amerindia MTDA, unlike the Eurasian sister haplotype, share informative S&Ps with Australo-Melanesians, Africans, or Neanderthals. These results suggest a unifying account of informative findings on the settlement of the Americas. So here's the introduction. Morphological analysis of early skeletons in the Americas have suggested that characteristics of some Pleistocene and early Holocene skeletons are different from present-day Native Americans and fall within the variation of present-day indigenous people in South Pacific, Australians, Melanesians, Polynesians, and Negritos, and certain sub-Saharan African groups. This is particularly so for the first South Americans, while the first North Americans seem to occupy an unresolved morphological position between modern South Pacific and European populations. No resemblance was noted between the first Americans and either Northeast Asians and modern Native Americans. This has led to the Paleo-American hypothesis that the initial pioneer population in the Americas had common ancestry with indigenous people in South Pacific, which was largely replaced by populations with Northeast Asian affinities in the early Holocene, but may have persisted in some locations in South America, such as the extinct Paraguayos and Fuego Patagonians. Archaeological studies have uncovered the Clovis culture, the oldest widespread archaeological complex to find in North America, dating from 13,000 to 12,600 years ago. The culture is thought to be developed in North America from an ancestral technology. Two competing hypotheses have been developed regarding the origins of the people who made Clovis tools. Based on the striking similarities between Soltrian and Clovis lithic technologies, the Soltrian hypothesis suggests that people of the Solitran culture, which was 21,000 to 17,000 years ago in Ice Age Europe, migrated to North America along the pack ice of the North Atlantic Ocean during the last glacial maximum. Alternatively, the more conventional hypothesis suggests that people associated with the Clovis culture were from Asia by way of the Bering Strait, and that the similarities with the Solitran tools are thought to be coincidental. However, neither the Paleo-American nor the Soltrian model has found support from past molecular resources. So it seems to me that they're saying that the Clovis, what they are crediting to the Clovis culture from Asia, may very well have been Solutrean, not Clovis. Just want to point that out. Ancient DNA studies on the 9,500-year-old Kennewick Man skeleton found in the state of Washington in the United States, which was thought to be closely related to the Anu and the Polynesians on the basis of craniomorphology, have nonetheless grouped him with present-day Native Americans. Moreover, populations such as the Fuego Patagonians that were considered to be relics of an early migration into the Americas and closely related to Australo-Melanesians are shown to be genetically related to contemporary Native Americans. Genome sequences of a 24,000-year-old Malta skeleton suggest that Native Americans are derived from a mixture of populations that are related to Malta lineage, as well as one or more unknown East Asian lineages. The 12,700-year-old Anzac 1 genome of the Clovis culture was also thought to be derived from Malta 1 and directly ancestral to many contemporary Native Americans. Why the dramatic differences between the molecular and non-molecular results? We have found the problem here to lie with the unrealistic assumptions of the present molecular methods. In fact, these assumptions have not even solved the long-standing puzzle regarding the determinants of genetic diversity. A new framework of molecular evolution, the maximum genetic diversity hypothesis, has recently solved the puzzle and made it now possible for the first time to infer molecular models of human origins based on genetic diversity data. So what he's saying, what they're saying is, they see the same thing that we see within this community. They're like, listen, we're looking at these skulls. They look like Negroes. We're comparing them to Native Americans. They don't look like them. We're comparing them to East Asians, which are ancestral to Native Americans, or at least they say. I guess you can say that's definitely true. They are ancestral to East Asians. But that East Asian type is not the modern East Asian type. But then again, it is the modern East Asian type, which opens up another can of worms because, like I said before, that East Asian type did not exist 20 to 30,000 years ago, at least in a modern sense. So when they say East Asian, are they referring to the modern type of East Asian, or they referring to the ancient type? I think in some studies they did say specifically an ancient type, but that ancient type is not going to look like, what Native Americans look like today. So, but this study is going to bring all of that together. But anyway, they're challenging the same thing that we're challenging. Like, listen, this don't make sense. We're looking at these skulls, we're looking at these skeletal remains, and what we're seeing is not consistent with what they're finding in the DNA, basically. And they're saying the reason why it's not consistent is the way that they're analyzing DNA. And they're offering an alternative method to better sequence this DNA. And this is what this article is going to be about. So let us read on to the next paragraph. It is now known that genetic diversities are mostly at saturation level, which therefore renders most of the past molecular results invalid, since those results were based on mistreating saturated phases of genetic distance as linear phases. Now, are these geneticists ignorant to this? Or are they purposely doing this to gatekeep? I'll leave that up to you guys to decide. Only slow evolving nuclear sequences are still at linear phase and hence informative to divergence time. For the mtDNA genomes, the relatively slow evolving sequences within mtDNA are non-synonymous sites in RNA genes, which are related to physiology and allow phylogenetic inference based on shared physiology. Let me read that again. For the mtDNA genomes, the relatively slow evolving sequences within mtDNA are non-synonymous sites in RNA genes, which are related to physiology and allow phylogenetic inference based on shared physiology. All right, new results based on the MGD theory have indeed suggested a unifying account of the origin of modern humans. The time for the first split in modern human autosomes was dated to be 1.91 to 1.96 million years ago, consistent with the multi-regional hypothesis. Modern Y and mtDNA originated in East Asia and dispersed via hybridization with archaic humans. Analysis of autosomes Y and mtDNA all suggest that Denisovan Neanderthal-like humans were archaic Africans with Eurasian admixtures who have transmitted their genomes mostly into the indigenous people in South Pacific. These new findings immediately make the Paleo-American model highly plausible. To address this and related models, we here reanalyze previously published DNA sequences based on the MGD framework. Now, from this point throughout this article, they're going to refer to this Denisovan Neanderthal-like human, archaic African with Eurasian admixture as solely Denisovan and Neanderthal. But you can see here in this paragraph, they're saying that it's Denisovan Neanderthal-like. And that they were archaic Africans. Now, we already know that in the West African population, that they were seen to have a ghost signal that was considered to be archaic West African, that was specifically not Denisovan or Neanderthal specifically. Said it was not like them. And as a matter of fact, it diverged before them. And that's how they know that it couldn't have been them. Now, this is saying something similar. I'm not going to jump the gun to say that it's the exact same type of signal, because this paper didn't go on to say that specifically. But I just wanted to point that out. And with me pointing that out, who is ancestral to who? This leads back to Albert Perry as well, with the A00 being found in his DNA, where they later found another lineage of A00 in Central West Africa, out of one of these DNA banks, which they were sitting on for a long time and never found A00 in them. And then all of a sudden, when they found it in Albert Perry, all of a sudden, all of a sudden they find it in Central West Africa, which is very peculiar. But we already know what time it is with the gatekeeping, But it seems that these black folks over here in America were actually ancestral to those in Africa and throughout the Western world, like we've been saying. And this article is going to continue to speak to that. But let me move forward. Results. Kennewick Man and the Paleo-American Model. We examine the relationship of Kennewick Man with present-day human groups in the 1,000 Genome Project. So once again, as I've said before, they pulled this DNA from, already existing DNA banks. So here you see it again, 1,000 Genome Project. People scoffed and laughed, like, you don't know what you're talking about. You see it again, the 1,000 Genome Project. This is where they get the DNA from, not random, samples from people. But anyway, in genetic distance by informative SNPs, homozygous mismatches in slow SNPs, as defined previously, Kennewick Man was closest to South American Peruvians from Lima, Peru, most distant to Asians, and about equally related to Europeans and Africans. I'm going to read that again. In the genetic distance by informative SNPs, homozygous mismatches in slow SNPs, as defined previously, Kennewick Man was closest to South American Peruvians from Lima, Peru, most distant to Asians, closest to Hunan peoples within Asians. But they're still saying that they were the most distant from Asians, and about equally related to Europeans and Africans. In contrast, two other ancient DNAs from North America, Anzic-1 and the 4,000-year-old Eskimo Sakic, were all more related to Europeans than to Africans. Okay? Anzic-1 was closest to Hunan and Southern Han Chinese, but Sakic was closest to the Japanese in Tokyo. Han Chinese in Beijing and Fujian populations among East Asians, indicating at least two different migrations of East Asians, with the ancient one by Southeast Asians consistent with modern human origin in Southeast Asia as recently discovered. Let me scroll this down. Recently discovered. These results indicate significant African ancestry in Kennewick Man, more so than East Asian ancestry, consistent with the Australo-Melanesian and African traits, interestingly, relative to PEL and Mexican ancestry from Los Angeles. I'll read it again. These results indicate significant African ancestry in Kennewick Man, more so than the East Asian ancestry, consistent with his Australo-Melanesian and African traits. Now, amongst the contemporary or mainstream academic world, it is circulated that the physical characteristics of Kennewick Man was more that of a European. But here, they're saying that he's European with heavy Australo-Melanesian and African traits, and his DNA is consistent with that. Yes, yes, detractors. It's on and popping. Yeah, yeah. It'll be no more of the you guys ain't, in the Paleo American DNA. You guys ain't got none of that. There's no Negro or African DNA in the Paleo American remains, in the ancient America remains. Not so fast. Detractors. Bring it on back, slow it down. Interestingly, relative to the PEL and Mexican ancestry from Los Angeles, Kennewick Man was not closer to Puerto Ricans from Puerto Rico and Colombians from Medellin, who are known to have recent African admixtures, suggesting very different African ancestry between Kennewick Man and the Puerto Ricans and the Colombians from Medellin, which is what that CLM refers to. We next studied the relationship of Kennewick Man with ancient DNAs of Neanderthals and modern humans. Because different ancient samples were sequenced at different coverages, it is unrealistic to use S&P mismatches to infer relationships, as there would be few shared S&Ps among different samples. As an alternative, we calculated the correlation coefficient R of 2 genomes and their distance to the 1000 genome project samples, assuming that different random sampling of a fraction of the whole set of 15,000 slow S&Ps are roughly equivalent in representing the whole set. We first verified this method by showing that the 6,000-year-old Iceman farmer from Italy was more correlated with the 7,000-year-old Stuttgart farmer from Germany than with the 45,000-year-old Siberian Ust-Ishim, and their distance to all 1,000 genome project samples. We also confirmed that a randomly picked CHS individual from the 1,000 genome project was more correlated in the average R values with the CHS group of individuals than with the Tuscany population in Italy, regardless whether the distance used in the correlation involved just one of the 5 major groups in the 1,000 genome project or the 1,000 genome project samples. Using the correlation approach, we found Kennewick Man to be most correlated with the 65,000-year-old Neanderthal Mezmaiskaya from Idagaya, Russia, among all ancient DNAs examined in their distance to all 1,000 genome project samples. In distance to only MXL and PEL samples, Mezmaiskaya 1 was among the top 3 most correlated with Kennewick Man, following just Anzac 1 and Malta 1. On principal component analysis plot, Mezmaiskaya 1 was located between European and Africans, similar to Kennewick Man. We have previously shown that Mezmaiskaya 1 Alta and VI 33.16 have more African than non-African genomes. These 3 Neanderthals all show far more correlation to Kennewick Man than Anzac 1 and Sakic, similar to the 4,500-year-old African Malta. But unlike the other Neanderthals, VI 33.25 and VI 33.26, who have been shown previously to be closer to East Asians and Native Americans respectively. All other ADNAs of modern humans examined here show more correlation with Anzac 1 and Sakic than with Kennewick Man. While the Denisovan genome also has been shown previously to be more related to Africans than non-Africans. So it says here the Kennewick Man is more related. They're basically saying that Kennewick Man is more related to modern Africans than non-Africans. It showed little correlation with Kennewick Man, consistent with its affinity to Australo-Melanesians. These results suggest specific Neanderthal-associated African ancestry in the Kennewick Man genome. We next ask whether some PEL individuals in the 1,000 Genome Project are like Kennewick Man and having more ancestry from Neanderthal-type Africans. Based on correlation with Mesmaiskaya 1 in distance to the 1,000 Genome Project, we selected 2 PEL samples, with one being top ranked and the other bottom ranked among PEL samples, and measured their genetic distance to the 5 major groups in the 1,000 Genome Project. These 2 PEL samples were about similarly related to non-African groups, but the most related Mesmaiskaya PEL HG02006 was much closer to Africans than the other. PEL HG01927 and more related to Africans than East Asians. This differential relatedness to Africans cannot be observed when fast SNPs and random set of 250,000 SNPs representing genome average, as described previously, were used to measure distance, consistent with previous failure to detect African admixtures using what are now realized to be adaptive SNPs. Now, I just thought of something, right? So we often relate, when I'm analyzing what is a Neanderthal signal or whatever, I'm finding out that too is, it's a title based on inference of, basically they title these genome types based on modern individuals. And so what they're doing with these Neanderthal types is they're saying, okay, this type coalesced like this, and we know that they are this much distance from what you would call modern human beings. But this is a title, you know what I mean? Because we're starting to find out that so-called Africans and Negroes have archaic related types of genomes that are very similar to Neanderthals. So what I'm saying is that is mainstream academia conflating what is African archaic with Neanderthals and Denisovans? And if that's the case, then it seems like Neanderthal and the Denisovan related signals within modern day populations like white Europeans, like East Asians, are actually archaic Africans. Maybe, just maybe. I might do a side, another in-depth analysis of that, to highlight that and go deeper into that. But for right now, I'm gonna continue with this study, but that's just some food for thought. Oh, and then also I want to mention as well, also with this, with the back and forth that I had with Sean P when I was doing the, African Americans or so-called African Americans are not West African. One of his arguments was that, you can't call, Australo-Melanesians Negroes like Black Americans, because they share these Neanderthal and really the Sonovan signals. They have about 5%, so therefore, their history is not the same as ours. But as it's starting to look, Sean Peasy, starting to look like that Neanderthal signal and the Denisovans signal is maybe either conflated or it might be misunderstood. How about that? But anyway, To further confirm the above results that the African ancestry in the PEL samples may not have come from recent African admixture, as in the case of that would be Puerto Rican and the CLM, we examined the genetic distances to the Thousand Genome Project for all American samples that showed close relationship with Africans as indicated by the PCA plot, including 2 PEL, 4 Puerto Ricans, and 4 CLM samples. Unlike the 2 PEL genomes that were far closer to American than to the non-American groups, and I think that would be Amerindian, and similarly related to African and Asian, the other samples also failed to show significant relationships with Amerindian relative to non-Amerindian samples, while were all closer to African than to Asians. These results confirm that the African ancestry in the 2 PEL individuals is different from those in Puerto Rico and the CLM individuals. The now extinct South American Fuego Patagonians are known to show Paleo-American cranial traits. We next tested whether these Fuego Patagonian genomes may be more related to Kennewick Man among the 3 ancient Amerindians. Relative to the North Amerindian Eskimos and Aleutians, Kennewick Man was the most related to the Pueblo Patagonians and Sakic than least related, consistent with expectations. We also examined ancient and present-day Europeans and found that ancient hunter-gatherers from Georgia or Caucasus were more related to Pueblo Patagonians than to Eskimos and Aleutians, whereas the opposite was found for ancient Western hunter-gatherers, farmers, Eastern hunter-gatherers, and present-day Europeans. Consistently, CHG was more related to the Neanderthal, Alta, and Mesmakia 1 among all ancient DNAs examined, while the Spanish farmers CB1334 showed slightly higher correlation with Mesmakia 1 than the CHG Coaches did. It was most related to the Ust-Ishim, whereas Coaches clearly had more Neanderthal genomes since its top 2 related aDNAs were both Neanderthals. We next used selected S&P's data from the Human Genome Diversity Project samples in South Pacific individuals as reported by Pugach et al. To further confirm Kennewick Man affinity with South Americans and indigenous people in South Pacific. We calculated that the ratio of R score of Kennewick Man versus ANZIC 1, as well as Sakic or PEL HG02006 or PEL HG01927 versus ANZIC 1 relative to Sakic and the 2 PEL individuals, Kennewick Man was more related to the Negrito group Mamanawa from the Philippines than to its non-Negrito neighbor Manabo, more to Amerindian groups than to others in HGDP, more to South American Sera than to North American Mayan, and more to African Yoruban than to Russian. In R scores, Kennewick Man was more related to Aboriginal Australians, Melanesian, and Manamawa than to the Han Chinese. Yeah, ain't gonna be no more of that, and none of that DNA found in ancient America. It's over. It's a wrap. We now have verifiable evidence from fellow geneticists. I ain't gonna say fellow because I'm not a geneticist, but from 2 geneticists confirming that the reason why these other geneticists are not getting these results is because of methodology. ANZIC 1 and the solar train model. We examined that the solar train hypothesis by testing whether ANZIC 1 may be specifically related to the 18,720-year-old El Moran from the Magdalenian culture in Spain that was immediately preceded by the Soltrian. In distance to Europe samples, El Moran was the only ancient DNA among all examined that showed positive correlation with the ANZIC 1, but not with the other two ancient Amerindians, Kennewick Man and Pt II. The correlation of other European aDNAs with ANZIC 1 likely indicates a general European element, but not special ancestry relationship, as they were also similarly correlated with the other two ancient Amerindians. The 7,300-year-old farmer CB13 from Spain was the most correlated with ANZIC 1 among European aDNAs, consistent with a special connection between El Moran and ANZIC 1 and local genetic continuity. As the number of informative slow SNPs available for El Moran was relatively small due to low coverage, we verified that they were good enough to show highest R value with Lebrana 1 among 25 ancient DNA genomes, as might be expected given that both were from Spain. Furthermore, among 230 ancient European genomes from 27 populations, 2 in the top 3 correlated with El Moran from Iberia. Further confirming the validity of our approach, we found, as expected, that the 2,400 or 24,000-year-old Malta 1 belonging to the Dravidian culture in south-central Siberia was more related to the 30,000-year-old Vastanis 16 of the same culture from the South Moravia of Czech Republic, and vice versa amongst all aDNAs of similar ages from Europe. Unlike previous findings of Malta 1 being closest to contemporary Amerindians, our results showed Malta 1 to be closest to contemporary Europeans, followed by highly admixed CLM and Peruvian groups, which is more consistent with expectations based on the location of Malta 1 in Siberia. What's also interesting about what they just mentioned is that Southern Native Americans show more affinity to these ancient fossils. You know what I'm saying? And I think that's going to come into play even more as we continue to read. We next asked whether the American group of 1,000 genome project may contain an individual specifically related to El Moran. Of all 352 MXL, PEL, COM, and Puerto Rican samples, we found one MXL NA19764 that correlated with El Moran and Lebrana 1, but not with 14,000-year-old Villa Bruna from Italy and the 30,000-year-old Vastanis. Interestingly, a significant fraction of the sample showed negative correlation with El Moran, while only a few samples did so for 26 other ancient European genomes examined. Among those MXL samples with positive correlation with El Moran, the average R value in El Moran correlation was similar to that for the Vastanis 16 correlation, indicating that a generally weak relationship with El Moran cannot explain the negative correlation with MXL. We further found that MXL samples with negative correlation to El Moran were closer to present-day Africans. I'm going to read that again. Interestingly, a significant fraction of the samples showed negative correlation with El Moran, while only a few samples did so for 26 other ancient European genomes examined. Among those MXL samples with positive correlation with El Moran, The average R value in El Moran correlation was similar to that for the Vestanese 16 correlation, indicating that a generally weak relationship with El Moran could not explain the negative correlation with MXL. We further found that MXL samples with negative correlation to El Moran were closer to present-day Africans, and Kennewick Man were more distant to El Moran and LeBron 1, with El Moran more distant than LeBron. This pattern suggests that El Moran related people, upon landing in North America, had admixed with African-like Amerindians who had settled earlier, therefore resulting in offspring with less El Moran and more African genomes. Oh yeah. Basically, they saying that it was a population group that came from Iberia that was more European related. But when they got to the American soil, those European signals got watered down by African related signals that they found here to already be here. This negative correlation of El Moran with a significant fraction of Native Americans was unique among the 26 ancient European genomes examined, suggesting that it was El Moran related population. It was an El Moran related population rather than any other group of Europeans that went into America and hence had their genomes partially replaced in their descendants due to admixture with African-like ancestors of Kennewick Man. Amerindians in the north had more individuals with negative correlation with El Moran than those in the south, consistent with where the Clovis culture was located. We further verified the special correlation between El Moran and Anzac 1 by using their distance to Amerindian samples in correlation studies. These results strongly suggest that El Moran related people were among the direct ancestors of Anzac 1. Given the location of El Moran and the associated Magdalenian culture, the results here are most compatible with the Solitarian model. So what they're saying here is two things. They're saying, just like I said before, that the Southern Amerindians show more ancestral affinity to Anzac 1 than do Northern Native Americans from North America. What they're also saying is that this El Moran is ancestral to Anzac. So I'm gonna read it again. We further verified that the special correlation between El Moran and Anzac 1 by using their distance to Amerindian samples in correlation studies. These results strongly suggest that El Moran related people were among the direct ancestors of Anzac 1. So Anzac had multiple things going on within his autosomes. At this time, we're looking at melanated people from Iberia being ancestral to Anzac 1. And this is something I pointed out before. I don't know if I've said it in my videos or not, but I've been pondering on this idea that modern Native Americans may legitimately share the unilateral markers with samples like Anzac 1 and so forth and so on. But just like I've said definitely in my presentations, is that does not reflect autosomal history or full ancestral history. And what Native Americans are actually reflecting is an indirect descendant relationship from these groups. But the issue is that the mainstream academic reports are also reporting that the autosomal signals are consistent with modern day Native Americans, which these two geneticists found questionable and basically found um, polar opposite results with the method that they used. Now they're going to get into the mtDNA haplotype results, which is also very interesting. Our previous study suggests adaptive evolution mtDNAs with nuclear genomes in admixed people, with indigenous people in South Pacific sharing more informative, slow-evolving SNPs with Denisovans and Neanderthals. Using the MitoWeb database, we examined slow SNPs and mtDNAs to confirm the above autosomal results. Kennewick Man carried the X2A haplotype that is only found in Amerindians and different from other X haplotypes common in Eurasians. The absence of X2 in Asia has been considered as evidence for the Solitarian hypothesis. We found 4 X2 associated slow SNPs, among which only 14,502 X2A Pacific, while 1,719 is present in Denisovans, some Australo-Melanesians, and others. 12,397 and 13,966 are not found in the South Pacific. However, the X2A Pacific 14,502 is found in P7 101 and S2 205 that are common in Australo-Melanesians. It is also frequently found in N10A 32 by 32, 32 of 32, and R8B 6 of 21 common in South Asians. N11 is found common 4 out of 5, found common in Chinese, and I guess that's the Han H42A, 10 out of 18 common in the Basque. These results suggest admixture between Solotrean migrants carrying X2 and the first Paleo-Americans or ancestors of Kennewick Man who shared ancestry with people in the South Pacific and Denisovans and Neanderthals. These results carrying X2. We also found other Amerindian Pacific mtDNA haplotypes to share more slow SNPs with Africans, Europeans, and Neanderthals relative to their sister haplotypes in East Asia. Of 5 A haplotype Pacific slow SNPs, only 8,027 in A2 Pacific and also present in L1C of Africans and some RO of East Asians, while the other 4 are not common in non-A types, indicating impact of Africans in the formation of the Amerindian Pacific A2 haplotype. Oh boy. For the Amerindian B2 type, there are 2 B2 Pacific slow SNPs that are commonly found in Europeans, 11,177 in U5B and 3,547 in the HV1B, consistent with effective admixture with certain European groups. Haplotypes C1, C4C, and D1 are highly Pacific to Paleo-Americans and Native Americans, but do not have defining slow SNPs different from those of other C and D types. However, they share more Neanderthal mtDNA alleles than the other C or D subtypes common in East Asians. These results on mtDNAs provide independent evidence confirming that autosomal links between Paleo-Americans and Australo-Melanesians, Africans, Europeans, and Neanderthals. Oh yeah. Discussions. The DNA results here are remarkably consistent with previous models based on morphological and archaeological findings. By linking Kennewick Man with the Neanderthal Ms. Pt I and contemporary Europeans and Africans, the puzzle of his craniofacial affinity with Austro-Melanesians now has an intellectually satisfying answer. The Pacific genetic relationship between Anzac I and El Moran provides strong evidence for the Solitron model. We have previously shown that among present-day people, Australo-Melanesians have the most ancestry from Denisovans, who were archaic Africans with Eurasian admixtures. The results here indicate close relationship of Kennewick Man with Neanderthals, but not Denisovans, therefore largely eliminating the possibility of a direct migration route into America via the Pacific Ocean for Australo-Melanesians in South Pacific, which is consistent with the stronger signal in the South American population versus North American Native Americans as well. So this article is bringing it all together, man. The Y chromosome haplotype C is common in indigenous people in South Pacific, Asia, Siberia, and North America, and belongs to the newly found mega haplogroup ABCDE that is closely related to Neanderthals and Denisovans, which is consistent with Native Americans sharing paternal ancestry with Neanderthals and Australo-Melanesians, which is often not talked about. Yes, Native Americans share ancestry with Australo-Melanesians. That is not disputed amongst mainstream academia. Therefore, data on all three informative DNAs consistently suggests that the first South Americans had archaic African or Neanderthal ancestry. This article was beating y'all up, man. Therefore, the data on all three informative DNA consistently suggests that the first South Americans had archaic African and Neanderthal ancestry. The timing of first entry into America by humans remains to be estimated by the future DNA studies. Several human sites in America, including the Topper site in South Carolina, United States, the Burnham site in Oklahoma, United States, and in Brazil and Chile, suggest that humans were in the New World as early as 30,000 years ago to perhaps 60,000 years ago. Now it goes up to 60,000 years ago. We know that the Native American, even based on the modern model, had not diverged 60,000 years ago. And 60,000 years ago, there was only melanated people on this planet, based on a little map that I shared in another presentation. I think I put that in that bonus footage for the Native American ancestry or whatever, This old age for the first settlement is consistent with the findings here that ancient Amerindians had more ancestry from southern Chinese relative to northern Chinese. The recent findings of modern humans originated in Southeast Asia and the African or Australo-Melanesian like genome in the Paleo-Americans or Native Americans that could only have come from Neanderthals. Also compatible with results here is the recent discovery of the 130,000-year-old human site in North America, indicating that admixture of Neanderthals and modern humans might have happened in America, which appears to be the more likely model given the absence of the mtDNA C1, C4C, and D1 haplotypes in Northeast Asia and Siberia that share more informative alleles with Neanderthals than their respective sister haplotypes in Northeast Asia and Siberia. Oh boy. I think that's what I just said earlier. If Neanderthals were present in Northeast Asia and Siberia, there seems a priori no reason that they could not find their way through the Bering Strait. We found the 24,000-year-old Malta 1 Siberia to be the most correlated with Kennewick Man among European ADNAs in their distance to Amerindians. This suggests that the ancestry of Kennewick Man may include Siberians, which is consistent with intuitive expectations. Malta 1 has previously been found to be most related to Amerindians rather than Europeans, but our findings here that Malta 1 was most related to contemporary Europeans is more in line with the common sense that local genetic continuity should be the rule rather than the exception. Indeed, morphological studies in general consistently find local continuity in traits and cultures. Malta 1 also shows a specific relationship with Bestony 16, both belonging to the same culture, Dravidian. Past studies, however, failed to see the relationship because they have used fast evolving SNPs that turned over fast in response to fast changing environments. Our results suggest that Southwest Europeans of 15,000 to 20,000 years ago, such as El Moran, had a special genetic connection with the Clovis people, Anzac 1 in North America, more so than East Europeans and Siberians of that age. Consistent with leaving Europe in ancient times, the genomes of these people are largely absent in present-day Europeans, which is unlike all other ancient European genomes examined here. Such results suggest that the part of ancestry in Anzac 1 that is shared with El Moran can only have come from Europeans who had come to North America by way of the Atlantic Ocean. Therefore, people of the Clovis culture appear to be the progeny of Europeans and the first South Americans. Ooh. So it's saying that it seems like they're saying that the migration came from the Atlantic, spread down to South America, and then went back north. And that is consistent with the genetics. What's also consistent is that the X haplogroup being found in the east of North America on the East Coast and not in Alaska, Canada, or the Northwest United States, which is very interesting. This is all coming together. You guys can go look this up individually, what I'm saying, verify what I'm saying. And if you can come with a better model, then show me. This explains the morphological findings that the first North Americans show traits in between modern Australo-Melanesians and Europeans. Our results also showed a fraction of present-day Native Americans to share more ancestry with Kennewick Man or El Moran. Some North Native Americans show replacement of El Moran genomes with Kennewick Man related African genomes, consistent with the first North Americans occupying an unresolved morphological position between modern South Pacific and European populations. These results cannot be found when using the adaptive fast-changing DNA sequences because present-day people living in the same location may share adaptive sequences despite different ancestry. It must be emphasized that any result must be held as uncertain at best until it has been verified by at least one other independent approach, unless the result is a logical deduction from self-evident axioms. Previous molecular results based on unrealistic assumptions have routinely contradicted trait evidence and common sense. In striking contrast, the result from our new molecular method, both here and in previous work, have consistently found unity between molecules and traits. We expect this intellectually satisfying pattern to continue to hold in future studies of other evolutionary puzzles. Crack that head, boy. What they say now? Africans? Black Africans? Even though, we know that's just the title. You know what I'm saying? But the biggest takeaway, the most important takeaway, is there was a Negro type that was present genetically within Kennewick Man. And they also pointed out that they believe, based on the genetic evidence, that there was a Negro type that they're calling Black Africans present in the Americas before the other ancestral components to Kennewick Man and El Moran arrived here. Yeah. So no more of that DNA talk, no more of, oh, you guys weren't here. There's no DNA of, Black Africans or Negroes in ancient America. No geneticist has ever reported that. It's over now. It all brings everything in full circle. The Australian signal in the Amazon population brings it together. X2 being found on the East Coast and not anywhere else, bringing it together. Oh yeah. There will be nowhere for you all to continue to hide with the pseudoscience. Your days are numbered. And with that being said, this is the end of part 2 of The Ghost of Kennewick Man. Can you dig it? Thank you guys. Like, share, and subscribe. If you like the information, leave some comments. And I'm out of here, man. Peace.